Hello students. Let's have a quick summary of sexual reproduction in flowering plants. Firstly, flowers are the organs of sexual reproduction in angiosperms or flowering plants. In flowers, there are two main reproductive structures, the Androecium and the Gynoecium. Androecium consists of stamens, which represent the male reproductive parts. A typical anther is bilobed, dithecous, having two thecae or compartments, and tetrasporangiate, containing 4 microsporangia. Inside these microsporangia, pollen grains are produced through a process called microsporogenesis. This process involves meiotic division to form microspores, which later develop into pollen grains. Pollen grains are the male gametophytes. They have 2 layers of walls, an outer layer called exine, made of a tough substance called sporopollenin with germ pores, and an inner layer called intine. Pollen grains may be in a 2-cell stage, vegetative cell and generative cell, or a 3-cell stage, vegetative cell and 2 male gametes when released from the anther. Gynoecium consists of pistils, which represent the female reproductive parts. The pistil has 3 parts: stigma, style, and ovary. The ovary contains ovules, which develop into seeds after fertilization. The ovule has a stalk called funicle, integuments, protective layers, and an opening called micropyle. Inside, there is a tissue called nucellus where a cell called megaspore mother cell undergoes meiosis to form a megaspore. This megaspore develops into the embryo sac, which is the female gametophyte. The mature embryo sac is 7-celled and 8-nucleate, consisting of an egg apparatus, 2 synergids and an egg cell, 3 antipodals, and a central cell with 2 polar nuclei. Pollination is the transfer of pollen grains from the anther to the stigma. It can be achieved by abiotic agents like wind and water or biotic agents like animals. After reaching the stigma, compatible pollen grains germinate to produce a pollen tube which grows down the style and enters the ovule through the micropyle. 2 male gametes are released, resulting in double fertilization. Double fertilization involves 2 fusion events. In syngamy, 1 male gamete fuses with the egg cell to form a zygote, which develops into the embryo. In triple fusion, the other male gamete fuses with the 2 polar nuclei to form a triploid primary endosperm nucleus, which develops into endosperm tissue that nourishes the developing embryo. The zygote develops into the embryo through stages like proembryo, globular, and heart-shaped stages. The ovary develops into fruit, and ovules become seeds. Apomixis is the formation of seeds without fertilization, which is common in some grasses and useful in horticulture. Polyembryony is when more than 1 embryo is formed in a seed. The most important point here is the concept of double fertilization, which is unique to angiosperms. This process ensures that the embryo and its food supply, endosperm, are produced simultaneously, ensuring better survival of the embryo. That's all, students. We'll meet you in the next important video. Until then, keep preparing. Thank you.